Great. Well, since everybody is silent, we might as well get started with introducing our speaker today, who is Mariana Bernardi Bif, who is a candidate for a faculty position in ocean sciences in the field of chemical oceanography. And Mari is coming up to us today from the Monterey Bay Aquarium Research Institute, or MBARI, where she's currently a research specialist and was previously a postdoctoral fellow. And Mari started out her scientific career in Brazil, where she did both a bachelor's degree and a master's degree at Universidade Federal do Rio Grande. And she published multiple papers from her work there, both her undergraduate work and her master's work, on both toxic algae and then non-toxic cyanobacteria, nitrogen-fixing cyanobacteria. She then came to an institution referred to as the Rosenstiel School of Marine, Atmospheric, and Earth Science. Not sure if anybody's heard of that. So she did her PhD here with Dennis Hansel, and I was just reminded that she was actually our first graduate student to fully go through the ocean sciences program, because the department was a new department and had new graduate programs after the divisions turned into departments. And so in graduate school she worked on, or in her PhD, she worked on dissolved organic carbon from a global standpoint, and she has continued looking at that global picture of ocean biogeochemistry in her subsequent work. And as part of that is also heavily involved in fieldwork, has done a lot of time at sea all across the world, I think, and is also going to tell us a lot about instrumentation development that is enabling kind of next generation biogeochemistry on that global scale. There's many things I could mention. She has received several awards for papers and talks, including the Student Speaker Award when she was a PhD student here. But I think I will go ahead and let her get to it. Welcome, Mari.
Zoom recording is not on.
Okay.
Good morning, everyone. I am Mari. I'm very happy to be here again. And today I'm going to talk about constraining the biogeochemistry.
Recording in progress.
Constraining the biogeochemistry of marine systems with BGC-Argo flows. Let's got it. Okay. So if you're familiar with the Argo floats, they are robotic equipment that we deploy all over the world and is measuring temperature and salinity. BGC floats measure some other variables that are chemical variables such as oxygen, nitrate, pH, chlorophyll fluorescence, backscatter, which is a proxy for particulate organic carbon in the ocean, and I'm going to talk a lot about this during my talk, and irradiance, which measures the light penetration in the ocean. We can pair these BGC floats with multi-platform data then to understand biogeochemistry of the world's oceans and answer some questions that the BGC float data cannot only understand by itself. So my talk is going to be talking a lot about how to merge BGC floats with multi-platform data to understand global marine biogeochemistry. Hillary already introduced me. So my background is in oceanography with a master's in oceanography, PhD in ocean sciences. And then after here, during my PhD, I started working with BGC floats, with the technology. And a lot of the technology is developed at MBARI. So I got a postdoc position at MBARI, and right after I got a permanent position as a researcher. At MBARI, I work for GoBGC and SOCOM programs. So GoBGC is the program that is deploying the US part of the BGC-Argo program that is deploying floats all over the world. And SOCOM project is more focused in the Southern Ocean. SOCOM project already has 10 years, and GoBGC is in its year 4. At MBARI, I do technology development. So I develop new chemical sensors to be put into BGC floats. I develop new methodologies to work with existing BGC float data. I also do science, because our lab receives all of the data from the floats that are deployed all over the world. I get the first dibs on how the float look like, the float data look like. I choose my favorite floats and I do data science. And I also do outreach. So I help a lot developing workshops using BGC float data, like the one we did last year. We have another one coming up next year, and it's free for early career students and grad students to join and learn how to use these data sets in their research. I am also developing an online course on how to use BGC float data sets, and it can be taken by everybody. So today, I'm gonna give a brief introduction on biogeochemical cycles under changing oceans, just so we are on the same page of what is biogeochemistry. BGC-Argo floats and multi-platform data. So how can we merge multi-platform data sets to better understand marine biogeochemistry? And then I'm gonna give you examples from my previous research using one float to constrain biogeochemistry, a few floats to constrain basin-wide biogeochemistry, and then a fleet of floats to constrain global biogeochemistry. Then we are going to dive into using floats to constrain the biogeochemical cycles under changing oceans. We know our oceans are getting warmer and more deoxygenated. So I'm gonna use examples of my ongoing research that is using BGC float data merged with other platforms to understand marine biogeochemistry of marine heat waves, ENSO conditions, and deoxygenated areas. Then I'm gonna wrap up with what's in the horizon. As these data sets are growing, we need to train the next generation of students to use the data sets to understand marine biogeochemistry, making use of this data that is freely available. And I'm also going to talk about opportunities for collaborations between other disciplines. So just starting with marine biogeochemistry. So the carbon cycle and the cycle of other elements are tightly coupled in the world's oceans. So at the ocean surface, phytoplankton over here takes up inorganic carbon and inorganic nutrients to build their biomass. And they do this by doing photosynthesis, and this is called primary production. From primary production, this biomass then is divided between particles, the particulate organic matter, and dissolved organic matter. The dissolved organic matter then can last in the upper ocean until it's transported physically to the deep ocean, while the particles can be exported through gravity or through zooplankton. So zooplankton, for example, eat those organic matter, and then they go to deeper waters, and then they respire, and then these particles are then transferred with these creatures. This is called the biological carbon pump. So constraining how much primary production, POM and DOM is produced, then we can also better understand how much of this carbon is available for export. And constraining these export fluxes are really important then to understand how much of this carbon is getting trapped far away from the atmosphere than to be a little bit protected from the ocean atmospheric fluxes. So primary production in the upper ocean, and then this biological carbon pump acts on physically transporting this organic matter out of the surface. But the oceans are changing, they're getting warmer, deoxygenated, and it's important for us to understand how this primary production and how these carbon fluxes are going to change. This is a model that is estimating that the biological carbon pump, or that carbon sequestration, is decreasing, and it's going to decrease by 2100. And these areas where the carbon pump is thought to be decreasing are areas that are under climate change already. So for example, we have more extreme warm events now, and this is the map of the most recent extreme warm events. And you can see that all of these areas are getting warmer, and they're getting warmer more frequently, and these warm events are lasting longer. So if we understand a little bit of the carbon cycle and the carbon budgets in these areas, maybe we can better constrain what is going to happen with the carbon cycles in the globes as the oceans are getting warmer and warmer. Also, oxygen minimum zones, or areas of the ocean where oxygen goes to zero and where a lot of the biogeochemistry happens, are also in these areas where the biological carbon pump ought to get weaker. In this sense, we need a way to monitor how the ocean is doing and how it's going to change. Right now, the only technology that can do this, monitor these chemical changes and these carbon budgets, is the BGC-Argo program. They provide the baseline to observe these changes in the ocean, and they are there, and they're going to be there for years, and using that data, we can then better constrain these carbon cycles. They have been deployed everywhere, so what you're seeing here are all of the BGC-Argo floats that have been deployed ever in the world's oceans. And in green, the ones that are still active. They're free and available in real time. So anyone that is interested in biogeochemistry can download this data set and incorporate in their studies. And the way these floats work, we deploy them using cruises of opportunities. Once they're deployed, they go down to 1,000 meters. When it's time to sample, they descend down to 2,000 meters. They turn on all of those chemical sensors over here, and then they start profiling up to the surface. When it's time to, when they are finished sampling at the surface, they send data through satellite to the scientists on land. And the scientists then start doing the quality control of all of these data over here. It is an international effort, so although I work with the US side of the program, other countries are deploying floats. What you're seeing here in green, in dark green, are the fleet that US deployed. We have other partners, it's not only MBARI, it's other institutions as well, including NOAA or ML. But other countries are doing a huge effort on deploying and quality controlling these data sets. This BGC-Argo array was inspired by the Argo success. So the Core Argo floats have been revolutionizing the way we look at temperature and salinity in the world's oceans, and have been observing how the ocean is getting warmer. This is the density of profiles collected by the ships up to this date. And pinks and purples are areas that are we have a very high density of data. Argo floats, the temperature and salinity only robots, have been increasing this density by a lot. So now we can observe regions where we couldn't observe before. And we can make maps like this of how the ocean is getting warmer. Floats are now the dominant source of biogeochemical data for some parameters. So I'm gonna talk a lot about nitrate, because nitrate is a very important nutrient for primary production. Remember, when phytoplankton pick up the inorganic carbon, they pick up a lot of nitrate in a certain proportion. Right now, this is shipboard data up to 2021, and this is BGC float data. So we need to take advantage of this data set over here if we want to better constrain biogeochemistry. We need the cruise data, and cruise data is crucial for us to make sure that our floats are sampling correctly. So one thing that we do is that once these floats are deployed, we match the sensor data with the bottle data from cruises to ensure that our float data is accurate. And this is just an idea of how accurate oxygen, nitrate, and pH is just by matching float data with bottle data. So it's really highly accurate. We can do a lot of research. You do not fear the quality of data of BGC floats, even though they collect data by themselves. And we use cruises of opportunities. So we are not funded to deploy floats. Floats are deployed using other cruises. GoShip, the US Repeat Hydrography Program, is a big partner. So we put our floats in their ships, and they cruise these very large areas of the ocean where smaller cruises don't go. They deploy the floats for us, and then we cross our data with their data to better constrain the accuracy of our sensors. And we take advantage of GoShip cruises in another way. So BGC floats don't sample every single parameter. So for example, the carbon cycle has the POM, the POC, the part of carbon of the POM, it is measured by floats, but DOC is not measured by floats. So we can match data from cruises that sample DOC in a multi-platform approach then. So when we try to constrain the carbon budget, what are the fractions of each carbon, we can use cruise data. I have been involved on compiling cruise data into databases DOM, DOC data into databases, and this makes it easier to pair than float data with DOM data. These databases then, I'm going to give you 2 examples of how we use BGC float data with DOC, DOM data to better constrain carbon cycle. And then this work over here was not developed by me, it was developed by my very good friend Ibin Huang. And he was interested in understanding how much carbon each of those fractions of carbon were being produced per year in each latitude of the Southern Ocean. So he used the entire fleet of BGC floats that are deployed in the Southern Ocean, and he could constrain the amount of POC that was being produced in the productive season per year by using the pH sensor and then a series of equations. He constrained PIC, which I didn't talk before because it's just a tiny fraction of the carbon budget, but we don't have the DOC. So he modeled the DOC, the only fraction that we don't have. And to be able to make sure that this DOC was accurate, he then compared with shipboard data. And what you see here is how much of each of these portions of carbon that are made in the upper ocean, they are produced in every latitude of the Southern Ocean. So for example, here in the higher latitudes, where phytoplankton has plenty of nutrients during spring and summertime, we have a lot of production of particles. When we go to lower latitudes, we started having more production of the dissolved fraction. And this is because as this area doesn't have a lot of nutrients, the phytoplankton keep photosynthesizing and producing these very small carbon fractions that are very poor in nutrients. So this increases the amount of dissolved fraction from the particulate fraction. So this is just an idea of how can you see all of this partitioning and only using BGC float data. And of course, pairing with the shipboard data to make sure that you're measuring and modeling correctly. In this work here, we did a little bit of something a little bit differently. We still use the whole fleet of BGC float data from the Southern Ocean, but we wanted to understand when phytoplankton incorporate carbon from the DIC and nutrients, looking at the nitrate, what is the proportion of carbon to nitrogen that they uptake? So we use the pH data from the floats to estimate DIC, and then we use the nitrate data from the float to pick up the nitrogen over here, and then we did a parallel, you just do carbon to nitrogen ratio. This talks a lot about, again, how the microbial community is behaving. If they have plenty of nutrients, this ratio is going to be lower because they're going to be incorporating this nitrogen into their bodies. When we go to the lower latitudes, again, as I explained it here, they become more limited by nutrients, and then this nitrogen gets lower. So this is float data, and what you're seeing here in black is exactly what I said, increasing the ratios in lower latitudes, decreasing in higher latitudes. We compare this with a model that uses float data, that uses shipboard data, the Takahashi model, and they see the same pattern. So it's very similar. But we don't see the same pattern in some areas over here, and we pose that it's because the Takahashi method uses shipboard data that is older than the BGC float data. So this might be a change in the ocean biogeochemistry by looking at these changes over here. We use then data from shipboard of POC to PON ratio and DOC to DON ratio to try to explain these changes. And the idea is that the particles have a different ratio, they're lower, and then the dissolved has a higher ratio because it's poor in organic nitrogen. And then we help like understanding this data set over here. But with one float, we can already do a lot. So using one float from the Northeast Pacific Ocean paired with remote sensing, we could constrain primary production. So this primary production over here from one float just highlights the spring and summer blooms collected by this float. We can then pick up the POC data from this float and calculate fluxes of POC that are sinking from the upper ocean, that biological carbon pump. And when we saw from this data set is that, for example, net primary production starts in the springtime and peaks in the summer, and the fluxes happen later in the summer. We can then use that same thing that is being used to partition DOC, POC, and PIC. And we see here that, for example, these carbon pools, the proportion of these carbon fractions vary over the year. So first, when the spring bloom and summer bloom kick in, we have more particles being produced. Later in the year, when there is nutrient limitation, we have more dissolved organic carbon being produced. With a few floats, we can start understanding ocean basin carbon production. So here I used only the backscatter sensor, the POC sensor, to estimate POC production over seasons in an area that is very poorly constrained. So this is the subtropical zone of the Southeast Pacific and the Southwest Atlantic Ocean. They're not very limited by nutrients, and there is little studies looking at this area. So using this backscatter sensor, I could look at the sensor profile, and I applied a technique that partitions the signal of the sensor into large particles and small particles. And what you can see in here is that, for example, small particles are present in different concentration than large particles. Small particles, these are particles, phytoplankton biomass. They have interannual variability. And what is very interesting is that large particles or assemblages of large phytoplankton are present only below the mix layer, highlighted by this dashed line here. So we can understand a little bit about ecology and a little bit of how much carbon is being produced by what fraction of the organic carbon. And then once these particles, they are produced, they have a certain, they have the chance to fall into the deep ocean. If we then estimate how much of this carbon is being produced in the upper ocean, and we estimate how much of this carbon is being exported away from the upper ocean, we can estimate transfer efficiencies. So the percentage of carbon that has a chance to be exported at deeper depths, to be sequestered. So what you're seeing here is the monthly transfer efficiency in the Southeast Pacific and one in the Southwest Atlantic. And if you follow these orange dots over here, you see that there is seasonality on the transfer efficiency and seasonality in the Atlantic side, but way more seasonal in the Atlantic side. And this has to do with how the mix layer behaves. It has to do with export due to the mix layer getting shallower in these months over here or deeper. So there is a lot of explanation, but what is interesting to see is that transfer efficiency can only be estimated monthly like this because of the BGC floats. And with many floats, we can estimate primary production. So primary production has been estimated using shipboard data, as you can see here. Very hard to estimate. It's measured in bottles that are collected in the ocean surface. And these bottles, then once you estimate primary production, or the amount of inorganic carbon that is taken by photosynthesis, this data can then be used in remote sensing. So when satellites collect ocean color over here, they can estimate primary production, but they need data to validate. They use this data over here to validate primary production. We estimated primary production using float data. As you can see, we have a lot of data and using the oxygen sensor. We have more coverage using these floats. And we estimated net primary production by knowing the amount of oxygen that is produced by phytoplankton per day. So we use this entire fleet as if they are observing the ocean at different times. And then the net result is always positive because phytoplankton are producing, they are building their bodies and they are producing and consuming inorganic carbon. So if we know how much oxygen they are releasing to the atmosphere, we know how much carbon they are incorporating. So using this estimate, we then estimated net primary production. We compared with time series programs, BATS and HOTS that have been estimating primary production for years, and they match pretty well. We then compared our net primary production by latitude with satellite data. We don't expect them to match completely because the data sets have different coverage. But when we integrate this primary production over the globe, we find pretty similar results. So primary production from floats were estimated as 53 petagrams per carbon per year. When we compare with what their satellites give to me, 51 to 52. This just tells me that the satellite is doing a pretty good job in relation to what the float measures. But now we have an idea of how primary production works right now. As these floats continue to take data, we can then go back and revisit these numbers and re-estimate primary production. Now, we are going to dive a little bit of what we can do in areas that are under nutrient stress, extreme climate events with rising temperatures and deoxygenation. So I am very interested in this area. This is the Northeast Pacific, and the reason why I was very interested in this particular area is because 2 marine heat waves happen one after another. And we happen to have over 10 years of BGC float data over there. The first marine heat wave lasted 2 years and it was intensified from an El Nino condition, an extreme El Nino condition. And I used the data set from nitrate from the floats to estimate organic carbon production. And I found that in the second year of the marine heat wave, we had very anomalous organic carbon production. The second marine heat wave was less studied than the first one, so we know a lot of the impact of this one in the ecosystem. This one is more recent, and we happen to have this second float data over here. We know that the physical forcings were a little bit different between the first one and the second one. So we were curious to understand what was the impact in biogeochemistry, but also link on the microbial communities that were producing those organic carbon. So were these marine heat waves changing the community composition that was driving organic carbon production and export? That was my question. The second marine heat wave in here, you can see that, for example, was very fresh, and that first marine heat wave was more salty. So that was very interesting. And this is collected by BGC floats. So this is just salinity anomalies. So my question was how changes in microbial communities during marine heat waves affected POC production and export. So for this, this is an in preparation manuscript. We are now tying the genetics. So I'm going to show you the biogeochemistry real quick. I used a multi-platform approach. I used over 10 years of BGC float data. I had POC data and all of the other sensors. I use remote sensing to correct my chlorophyll data. So when I estimate POC, I like to tie with chlorophyll just to pinpoint the production. And the chlorophyll from the sensors need a little bit of more quality control. So I match remote sensing with chlorophyll and I correct my sensors chlorophyll. And this area happens to have one of the oldest time series programs in the world, this line P. So I use data from line P, cruise data, seasonal data that have been collecting data before, during, and after the marine heat waves. And we reprocessed their leftover water for genetics. So I could pair the locations where my floats were with the bottle data from the closest station to then analyze who were the producers at those different seasons, who were the zooplankton that were growing there. Was there something anomalous going on during these heat waves? What I'm showing here is BGC float data, and this is the entire time series from 2010 to 2022. These are the 2 marine heat waves, and the first thing that you can notice is the amount of particles that accumulated in the upper mesopelagic that is very different from other years. So this was very anomalous. So this was already pretty interesting. When we zoom in these 2 years of marine heat waves, we see that these particles were produced during the springtime. So in these 2 years that we had marine heat waves, this POC was being produced in the upper ocean and it was being exported and it was resisting degradation. It was accumulating in the upper mesopelagic. And then it was being gradually respired over the year. So why is this? We know from this recent exports campaign that the POC, the exports campaign, was a very big project that went to this area, and with cruises and sampled a lot of parameters in this area just to characterize the biological carbon pump. They found out that POC export is strongly tied to vertical migration. So when you see particles falling into the deep ocean, you would imagine a decrease in particle concentration, because as these particles are falling, they're being respired by bacteria, right? This is not what happens, and this highlights the vertical migration by zooplankton. So zooplankton basically feeds during the night in the upper ocean, migrate to the mesopelagic, and then they do their metabolism over there. They poop, they have interactions, and then in here, smaller zooplankton, they are going to eat up those small particles, and then this highly recycled system tends to have smaller and smaller and smaller particles that are then very hard to export. These anomalies are only seen in the small fraction of the POC. With that other technique that I showed you before, that I partition large and small particles, these are just seen the small particles. So we think that the zooplankton is exporting, and then these small particles are getting reworked. They tend to be too small to sink, so they are lasting in the mesopelagic. So the take-home message here is that export of particles was intensified by vertical migration during the marine heat waves. And now we are tying with the genetics. Our genetics are already done, and I can already tell you that during those marine heat waves, copepods changed composition. So we found very warm water copepods. We also found very small protozooplankton that showed up in those 2 marine heat waves in the upper mesopelagic, and they were never found there before. So those new species are changing the way that POC is being respired back to DIC in the water. But this is only one marine heat wave. We can do a lot of similar things in other marine heat waves. We have floats all over the world sampling. They already sampled marine heat waves. They are going to sample more marine heat waves. And as this phenomena gets more frequent and more extreme, we need to constrain other regions too. So now we are just putting a little bit of the genetics into our paper, but we have this huge amount of metagenomics that was created from the reprocessing of these samples. And this gives a lot of opportunity to write proposals using this data and pairing with BGC float data to better understand what were the drivers of the changes in the microbial communities. Just correlating these changes in the metagenomics with, for example, temperature, salinity, water mass densities, mixed layer depths. So this gives us a very big canvas to write proposals and have students to work on these data sets too. But not only marine heat waves warm the ocean, also El Nino conditions warm the ocean. And this is my second interest in looking at how changes in ENSO conditions affect marine biogeochemistry. I'm especially interested in the Equatorial Pacific, where we recently deployed floats, and these floats have been capturing the water properties from before this last El Nino that just finished and during this last El Nino. So my inspiration for this work is because a few years ago, I used GoShip data, that repeat hydrography data, for a paper, Constraining Biogeochemistry of the Upper Ocean. And I saw that nutrient concentrations were different at different occupations of the same line, of the same cruise line, between 1994, 2008, 2017. And in La Nina conditions, you have higher nutrients in the upper ocean, around the Equatorial Pacific up to about 15 degrees south. During neutral conditions, you have lower. But what happens during El Nino conditions? El Nino conditions are thought to restrain the vertical mixing with the upper ocean. So you would imagine that primary production would get a little bit lower because they have less nutrients. So I'm just going to show you a glance of how our BGC float data looks like. So this half here is before El Nino. This is during El Nino. And this floating here sampled the equatorial zone. We see that chlorophyll starts becoming a little bit less concentrated, so you have a little bit less phytoplankton growing. And nitrate, that nutrient that is key for primary production, becomes more limiting. We don't know that if it's because of El Nino or if it's because of physical transport to areas of different water masses. That's the detail. When we look at these floats over here that are more around 10 degrees south, we see that chlorophyll gets way lower during El Nino conditions. And then when we go even further south, and this float didn't really move that much, we see something opposite happening. We see that chlorophyll becomes more concentrated. We have more phytoplankton growing. And nitrate becomes less limiting. So that was very interesting and very counterintuitive. And we are going to explore this area more. I have a summer intern coming to work with me. She is a grad student from Scripps, and she's a physical oceanographer. So we are going to be constraining this, the mixed layer depths and how mixing can influence these different biogeochemical parameters over here, and then also looking at the vertical, the horizontal transport of this float versus if it's a horizontal transport or if it's an El Nino condition. And then this is my third example of using float data to look at extreme events. And what I'm saying here is that oxygen minimum zones are expanding, and they are very important for biogeochemical transformations, and I am going to explain why. What I want to point out here is that these areas in purple are areas where oxygen falls near zero. And in these areas, they are thought to be expanding, and this is the area that I'm very interested at. So basically, when we see particulate organic matter falling into the mesopelagic and in that subsurface, we have bacteria respiring this organic matter back to inorganic carbon, and they use oxygen for that. Once they run away of oxygen, they want to keep eating, right? So they find other chemicals that they can steal the oxygen from. And particularly nitrate is a very important nutrient for bacteria. So they pick up organic matter, and instead of picking up the free oxygen that doesn't exist anymore, they pick up nitrate. And they start reducing this nitrate into nitrite and then into gas. This gas then escapes to the atmosphere. So we lose nitrate. So these areas remove 30 to 50% of the ocean's fixed nitrogen, but they correspond to a very small area. Understanding these reactions over here are very important because they are made by different microbes. And understanding the proportion of these different reactions is very challenging. It's very hard to go there in cruises, sample that data that doesn't have oxygen, with very careful not to contaminate your samples, and then measuring these microbial reactions here. So there is a way to measure the proportion and the importance of these reactions at different layers of the ODZ using BGC float data. But as you can see here, nitrite is a key part of all of these processes, and we don't have nitrite in here. However, this is my ongoing research. I am using the output of our nitrate sensor, and I am reworking the signal, the output that it gives to me to analyze nitrite and also other sulfur components that are very important in the oxygen deficient zones. So sulfur as sulfate, SO4, is also reduced in the oxygen minimum zones, and they're also part of these the biogeochemical cycles in there. So basically, this is my nitrate. This is the head of a float. This is what I receive in the lab. And we put all of this together. This is the nitrate sensor. This is my spectrometer, my lamp, and the probe. And when we put all of this together, this is basically a spectrometer. When we throw this nitrate sensor in the water, it's not really a nitrate sensor. It's a spectrometer that measures a UV range. It happens that in most of the ocean, the UV, the only component other than salinity that absorbs in the UV spectra is nitrate. So then we deal that as nitrate. But when we go to the oxygen deficient zones, there are other components that happen to absorb in the UV range. So I characterize these components in the lab, and I basically use a different statistical approach that takes into consideration a known absorbance of each of these compounds from the ODZ. And then I use a statistic that fits this data and finds the probability into concentrations that I'm gonna find these certain components. So for example, it looks at the shape of the sample that the nitrate that the sensor is giving to me. If there is only nitrate, it's gonna give me a shape that corresponds to the absorbance of nitrate. If I have nitrate and nitrite, it's gonna give me the combination of these two shapes that is defined by their own original shape. So I can reanalyze this output, and then I can give you nitrite concentrations. One way to prove this is by going in the sea, sampling nitrite, and then matching with my new approach. And we did that. So we used data from a cruise that came in the Eastern Tropical North Pacific ODZ, sampled nitrite using bottle data. We used their data set and we reanalyzed this float data, and we have a pretty good match. So this is nitrite from floats in red, in black from cruises. And you see that they match pretty well. So these are only the first few profiles, because then the float goes away. It's another system. But they match pretty well. The beauty of this method is that we can reprocess all of our BGC float data, and we can get nitrite from everywhere in the world. This is not very easy to measure. Shipboard data don't really go and measure nitrite very frequently, and not at very high vertical resolution. This is a very high vertical resolution nitrite, and then we can start looking at the structures of the nitrite concentrations. And if we put in our model, that we used a model from a paper that look at nitrite concentrations and other parameters in a cruise 2 years before this one, and they could define those reaction rates. If we use that model using our float data, we can define the reaction rates of microbes at different regimes. So this is a time series of nitrite for the first time every 10 days for 2 years. And you see that there is a high seasonality. There is a structure in the water column. And this cannot be seen by just going there and taking a snapshot of a profile. We can see that this nitrite actually, when microbes reduce this nitrite and also remineralize the nitrite back to nitrate, some reactions are very tied to inorganic carbon. So for example, in areas over here in the upper ODZ, you see this high pH forming. This has to do with the biogeochemistry of microbes. And here is just to show you that in this area, oxygen is lower than 1 micromolars per kilogram. So this is pretty low. This area also has to do with, this variability has also something to do with POC export from the upper ocean. So this is integrated POC for the upper 100 meters. You see that POC has a little bit of seasonality over there. In this first half of the time series, it is a little higher. You see a big export going on. This is also tied with these transformations. So I'm just going to give you a glance of what happens when we put all of these data sets into the biogeochemical model that looks at the different reaction rates of microbes. And this is what we see. So we divided our time series between low nitrite and high nitrate because we believe that different biogeochemical reactions are happening, right? And what you see here are just 2 reactions as an example, denitrification and NMOX. Both of these reactions transform nitrite into N2 gas. And what we see here is that these 2 reactions happen at different layers of the ODZ. The ODZ is right here. You see the NMOX is more homogeneous, and then denitrification happens more in the borders of the oxygen deficient zone. This has to do with PO particles falling. It is a whole story that we can tell with these reactions, but it's super interesting. And then I told you that we can use this data to understand sulfur cycle. So another component that we can measure is thiosulfate. That is an intermediary compound between sulfate reduction to sulfide, the S2, that also is lost to the atmosphere. It becomes gas and it runs away of the ocean. This is thiosulfate. You see kind of like a similar pattern, and we are still exploring this data set. We are still validating this data set. So I hope with these examples, you could see the power of the BGC floats, what we can use, how can we understand different biogeochemistries, organic carbon production, the different partitioning, a biological carbon pump. We can tie with genetics, we can tie with physical oceanography to better constrain that production. And there are many opportunities to investigate marine biogeochemistry using these data sets. Everyone from basically any discipline can incorporate this biogeochemical float data into their own discipline to then maybe enrich or answer a different question. There is a lot of opportunities to train students on the use of these data sets. There are a lot of things that we can do together. For example, NOAA-AOML has a relatively newly formed group of people working with BGC-Argo floats. So it will be very interesting to work together with them, developing new technology and looking at the science questions. There is also opportunities to develop technology in the lab. So this is only one example. This didn't take a development of a new sensor by itself. It was just a new technique, a new way of looking at the sensor's output. But as these floats are being deployed in the oceans more and more, there is need for better sensors, cheaper also in relation to power, because power is a big problem in BGC floats. So developing sensors that are a little bit consume less energy. There is a lot of opportunities to develop new things. Also, again, train the next generation of professionals. And the beauty of this data set is that students can use at different levels. Undergrad students can download this data set over here and throw this data set into an ODV program, Ocean Data View, and take a look at time series. As they learn how to download data and look at the ocean's data, they learn about the ocean system too. So students can learn how to process and quality control this data at undergrad and graduate level to grad school. As they take the fear out of big data set, they can download basically any data set. If a student can learn BGC flow data, they can then start downloading remote sensing data and pairing with buoy data and pairing with shipboard data. As students are familiar with this data set, they start understanding the little changes in the ocean, how seasonality plays a role. They can look at the nutrient regimes, they can look at how temperature and salinity changes, how density changes. So it's a very rich data set to train students on understanding ocean sciences. And we already have toolboxes that are written in MATLAB, in R, and Python that anyone can download from our website. And we can use these scripts. And these scripts are going to basically give you, you can tell which area of the ocean you want to download the data from, which variables, only good data, not bad data, I want only good data, only in the Southern Ocean. And then they download the data for you and they make basic plots. So students or even scientists that want to incorporate these data sets can use our toolboxes to download data and start playing and looking at the ocean data in your favorite area of work. My dream is to use this data set to teach. And I want to use BGC float data in a course that teaches data science of marine systems, that is going to bring together students from different years in only one class, and they're going to learn in their different levels of skills how to download the data set, process data, and develop projects in ocean biogeochemistry. So for example, a data where students can first pick their favorite area of the ocean and stick with that area for an entire semester, working and developing tools to understand the biogeochemistry of that area. This was inspired by a talk from Scott Glenn and Oscar Schofield. They have been implementing a class like this using other data sets in Rutgers, and they have been very successful for over a decade. And I think that these would really take, this is a really a very new take on how biogeochemistry can be taught and how the use of ocean data sets can be taught. So students can learn from acquiring data, merging with other tools, other platforms. They can process data, quality control, and then for grad students that are interested on answering questions of biogeochemistry using float data, then having them published as papers. So this is just if I could summarize what I want to do here. I want to bring my experience with the BGC-Argo array and other observing platforms to UM and guide students in their path to understand and quantify the biogeochemical cycles of our changing oceans. So I feel like it's always fun when we involve students. I think it's very rewarding to see people learn, and I would like to expand my circle by teaching people, not by doing very high-level science by myself and with my very expert friends. So I would love to expand this to students. With that said, thank you. Now take questions.
Yeah, we have time for questions. I think just if you have questions from the room, just maybe repeat it so people on Zoom can hear, and vice versa.
Mm-hmm.
Can someone repeat the question, please?
I will.
Yeah, you just something you mentioned, the Gulf of Alaska. You talk about nutrient limitation coming in and then changing the proportionality between POC, BOC, and all that sort of thing. So, but before that was, the nutrient limitation would be nitrate, but up there would be iron. So, does it kick in the same way? Does it have the same impact to be iron limited as to be macronutrient limited as far as the change in the proportionality?
So Dennis asked me, because I talked about nutrient limitation by nitrate, and he asked what about iron, and if I can tell anything by using BGC float data. That was the question. It is probably going to be a little bit different. Let me show you an example here. We cannot, it's our dream to make an iron analyzer, just putting right there. But these two areas perfectly summarize what happens. So Southeast Pacific is known to be iron limited. Southwest Atlantic, not so much, because we have a lot of atmospheric inputs, and continental shelf from the Patagonia. And we have more particles being produced in the Atlantic Ocean versus in the Pacific. When we look at the particle concentrations, POC, production per month, is higher in the Atlantic than in the Pacific. So this gives us a hint of what iron limitation can do, because two areas at two different latitudes could be producing similar POC concentrations, but they are not producing because we know that one is limited by iron and the other one is not. So you can tie with existing information. So this is just an example. Yeah, we cannot go, we will need to go there and measure, but yeah. Another clue is on the chlorophyll signal. So I don't show you here the data of chlorophyll, but chlorophyll measured by floats is chlorophyll fluorescence. It's a little bit different from real chlorophyll. When we pair with remote sensing, when we match the chlorophyll that the float measures with remote sensing, there is a ratio that changes. And this is thought to be iron limitation, actually, in the Southern Ocean. This was done by Christina Schallenberg in Australia. She's an amazing scientist in Australia. So looking at those ratios, you can kind of talk about iron stress. So I use that data to correct my chlorophyll from this work. I'm just not showing here, but you can play with that data too. And it's very seasonal. You actually see the seasonal changes in these ratios. It's very interesting. Yeah? Any more questions?
Hi, Mariana. So I'm very interested in the diurnal variability of sea surface temperature. And so I saw in one of your slides you had some results where you looked at oxygen variability as a function of the local time of day.
Yes.
And I'm just wondering, is there anything in the Argo floats or the BCG floats that sets like the time of ascending?
Yes.
Or is it completely random? How does that work?
Yes. So this work was inspired by a paper that looked at diurnal variability of temperature from, she's in Scripps, physical oceanographer.
Janet? Lynn?
Not Lynn.
Janet?
No.
Sarah Gilly?
Gilly.
Yeah. She
Gilly's
was my
paper.
advisor at Scripps.
She took all the Argo data and she did the diurnal variability of temperature, and it's beautiful. So when we did this work, we realized that a lot of floats were sampling at the same time of the day, usually noon, so they could capture the chlorophyll, maximum chlorophyll of phytoplankton, and we had to toss out the samples because they would be biasing our analysis. So then Ken, the principal investigator that I work for, asked, please people program the floats to sample at different times of the day so we can do something like this. So now with some types of floats, we embedded in the code a few minutes. I don't know if it's a little over 2 hours. So the float have a little lag every time they go up and sample, so we have the uneven distribution. For some other floats, the code is hard-coded, so you can't really change from land. So those floats, you kind of need to change like manually before they sample. It's a big hassle. But we are trying to make all of the floats that are always deployed, ever deployed, to sample at different days.
But so wait a minute, but like you say it's hardwired so that it's at noon, but doesn't that depend on the longitude? So
no, the way the software is designed is that at a certain, every 24 hours it turns on.
Okay.
And then I think they code the float a little bit before they deploy to tailor it to every area that is being deployed. So they hard coded everything for noon, and not everything. Thank God it's not everything, because otherwise we couldn't do this. But we tossed a lot of floats. Yeah. Any other question? Yes.
So if you had your pick, I guess you already hinted at it, what other sensor would you put on the Argo floats?
So Moha is asking what other sensors I would put in an Argo float. Iron, DOC, DOC for sure. I would also develop a nitrate sensor that uses less power, because the nitrate sensor, because it has a lamp, is the one that draws the most of the power from float. And if we could, if we could make these floats live longer or sample more frequently, it would be amazing. Nitrate sensor draws about 40% of the power of the float. But it's so powerful for so many things that we can constrain that we don't want to, it's a key. But yeah.
What about ammonia and silica?
They tried to make an ammonium analyzer, and it's usually using microfluidics. The problem is that the sensors cannot hold chemicals because it's very risky for the float. It will change its volume. So everything, it would be amazing if we could have an ammonium analyzer or silicate. And I think there are ideas also for...
Silica has an spectral signal.
Yeah, so these are things that can be developed. The only thing that we are limited by is that we can't do chemistry in the sensors, right? But it would be amazing, yes.
So it's a more general question. You were thinking about like visions and everything. So I'm a bit ignorant of like the relative importance of like coastal areas versus the deep ocean when it comes to like global carbon. So obviously with the BGC-Argo, you don't access those coastal areas, right? So if you think about global budget, are we missing a lot by not having the coastal areas? And so how do we deal with that in the future?
So Shane is asking, what about the carbon budgets in the coastal area? Because we don't sample with BGC floats if we are missing a lot of that. And my answer is that again, the idea of using multi-platform. There are a lot of people using gliders and sea drones, and they are meshing sea drones and gliders with BGC float data to then better constrain the carbon cycle. Yes, we are missing a lot, but a lot of the carbon production in the shelf is shallow production, so it doesn't tend to export real deep unless it's transported. Of course, we are missing a big puzzle, but it is very important to understand the coast. The problem is that the floats are not designed for that. So meshing with gliders, meshing with sea drones, this is the beauty of meshing multi-platform data, right? Yeah. Any other question? Was it?
Yes, maybe it was done, but I noticed that most of the analysis that you are doing looks like more like you are working with some equipment just fixed on a place, so I don't think you are taking advantage of this Lagrangian Argo characteristic. And I wonder, because I mean, they cover a long area. I think you are limiting yourself to a specific area to do the analysis, but I think for fluxes, there should be, the advantage of being Lagrangian has to be.
Yes. I have been using a lot of floats that tend to be very confined, and the Lagrangian approach is what we are trying to use to look at the El Nino, because this is a perfect example, because the float is traveling super fast in the equator. Okay. So is this the effect of El Nino, or is this the effect of just being transported to an area where it has less nutrients, right? So again, the importance of working with physical oceanographers and communicating with each other, right? Because unfortunately, I'm not a specialist in every field, so it's the beauty of collaborating with people. Like my grad student that is coming, she works with Lynn Talley and Matt Maslow, that are biogeochemical modeler and a physical oceanographer, to exactly try to constrain this horizontal advection, versus the biogeochemical cycles.
My other question was related that, not because of the Lagrangian advantage, but also you like checking if you end up in an eddy in a front, or you are in a very regular area, or these kind of things, that for sure will change your characteristics. Yes,
so Josefina is talking about when these floats travel, they face eddies, mesoscale processes, and I am going to comment on that we are looking at that actually here. In this study here so this study here in the oxygen deficient zone is actually an eddy that pump the particles in here, and we are working constraining those eddies that are in the region. We are also looking at the omega values, so the dissolution rates of calcium carbonate and aragonite, and they correlate very well how shallow and how deep they go with the passage of eddies. So in this work we are going to talk a little bit of submesoscale processes, because this is a particular region where eddies happen. This float didn't capture too many, but we have other floats more south that capture a bunch of eddies, and it's pretty interesting. So again, just expanding what we can do, right? For sure, we are worried about this. And for example, when we analyze those carbon budgets, we take into consideration advection and vertical transport. We do a very truly carbon budget assessment. Yibing Huang is a very amazing person that I collaborate with a lot, that does a lot of these assessments of the advections playing a role in the biogeochemical concentration of the chemistry. Any other question?
If not, let's give Mari our thanks again. And,
can we ask online?
Online questions.
Oh,
Yeah,
sure.
thank you for your nice talk. So as you emphasized, this Argo floats dataset is a community dataset and it's globally used. A lot of people are using it So I'm wondering if you can emphasize what will be your niche, what you do, what's your edge, what you do better than others, or and also, do you plan to work on this data set for the next few decades, or do you have other plans, kind of thing?
Yes. So I don't think I do better than anyone. I think I have my favorite areas that I like to focus in, and here what I am focused on is looking at areas that are under change. So areas where we see El Nino, La Nina, ENSO conditions, areas that are under marine heat waves, which are many, oxygen deficient zones, continue to constrain the nitrogen cycle inside oxygen deficient zones. There are so many areas. We are going to the Arabian Sea, hopefully, if security allows, next year, to sample in the Arabian Sea. That's an area where we don't know anything about the oxygen deficient zone. So my plans are to continue working on the sensitive regions here for the next mid to long term. And well, the ocean just got a break record on temperature. And there are very scary articles saying that it's gonna get even warmer than we predict. So I guess my areas are gonna expand with the years. So this is my idea, to continue working in these areas. And my view is a very system view of the ocean. I'm not a specialist on pH or nitrate, so if you talk to a pH specialist, he's gonna be better than me for sure. But I like to work with all the sensors in a very integrated way. I'm not afraid of collaborating with people that can help me answer the questions that I'm interested in. And I feel like this is what motivates me in science, is to talk to people that can do things that I can't, and then we can do something very cool together. So that's my answer for that.
Thank you.
There was just a question. I think the units might have been weird on one of the slides, is what that question was about.
Claudia, very low oxygen.
I think the units are off because...
Oh, yeah. Sorry, the microgram, yeah. It's very low oxygen.
But not that low, because the sensor is plus or minus 3 micromolar, right?
No, it's
I think that's what you showed early.
0.1.
You showed 3 micromolar. On her
Yeah,
That's the
the
detection.
accuracy, but the limit of detection is 0.1. So yeah, it's less than 1. It is not incorrect actually.
Well, it shouldn't be that low at the surface though.
Oh, sorry, it's very blown up in here, so we can identify the structure over here. But at the surface is way above 1 micromolar per kilogram. Yes. I just blew up so we could see the structure of the oxygen deficient zone in this area. Yeah. So yeah, it's correct. There is more oxygen than 1. So everything that is black is bigger than 1 micromolar per kilogram. Does it answer you, Claudia? Yes. Okay.
All right, we will now let everyone go. Thank you again, and
Thank you everyone in the online.